Perfect. Okay, thanks again. So we're going to begin our discussion of cellular respiration by talking about ATP, because ATP is our body's main currency, if you will. Okay, it's going to be what we store up our energy into this form, especially in this third phosphate. A lot of energy is captured in adding that third phosphate onto the two existing phosphates of ADP. And so this is how we put our energy away. And I like to refer to this as a backup battery, right? You charge that backup battery, you put it in your bag, or you keep it on your person, and you have it as an additional energy source. That is the same for ATP. Okay, now it's going to take exergonic reactions, right? Exergonic meaning that energy is going to be stored into something. Energy is going to be taken or reserved for later use. And so that is how we're going to make ATP. All right, now, how do we actually do this? We have ADP, which has two phosphates, and we have the extra phosphate, which again is storing lots of energy. We're going to combine that, and then because this is a hydrolysis reaction, we're going to release water, right? Excuse me, a condensation reaction, we're going to release water, and we're going to make that ATP. Now, why do we do this? This is how we can harness the energy from the food that we eat. So in our last chapter, we talked about carbohydrates, fats, proteins. We talked about them being some of our body's main source of obtaining energy. And so we're going to take that energy. We have to be able to harness it. We have to be able to capture it and store it in our body so that we can use it to do cellular processes or cellular work. So we're going to liberate that energy. Again, this is going to be an endergonic reaction to take the energy, right? About 7 kilocalories per mole. And because this is a condensation reaction, we're going to add that energy and we're going to release water and make ATP. Now, there are two different ways that we can make ATP. And this is basically phosphorylating that ADP, adding on that third phosphate. And that is one substrate level phosphorylation, which is basically getting a phosphate onto ADP, but obtaining that phosphate from another molecule. I like to refer to this as borrowing a phosphate, right? Or stealing a phosphate, if you will. So molecule X here has a phosphate on it. ADP is going to pretty much grab that phosphate from molecule X. And on the right side of the equation, we're going to have molecule X, which is no longer with the phosphate, and then the phosphate on ADP, now making ATP. All right, so that is substrate level phosphorylation. We typically do this in the cytosol of the cell, and I'll talk about this some more going forward. And again, this is analogous to borrowing or taking a phosphate from another phosphorylated molecule. The other way of making ATP or phosphorylating ADP is oxidative phosphorylation. And this is a really important, probably the most important source of us making ATP. It's going to require the electron transport chain, and it's going to require oxygen, right? That's kind of implied in the name here. So we've got oxidative phosphorylation, meaning oxygen must be present for this form of phosphorylation to occur. And this has to happen via the electron transport chain, which is going to be in a different part of the cell. This is going to be in the mitochondria. So just to recap here, substrate level phosphorylation makes us a small amount of ATP. We're going to borrow the phosphate to make that ATP. This is going to happen in the cytosol, right? And oxidative phosphorylation is going to necessarily require oxygen. This is going to happen in the mitochondria, and it's going to require it's going to make or generate a lot more ATP than substrate level phosphorylation. Really important concepts to be able to differentiate, because we're going to speak about these here in cellular respiration, and then we're going to revisit them again when we talk about muscle in chapter 11. Or chapter 12. So it's really important to understand the differences in how we can make ATP. A small amount of ATP very quickly in the cytosol, or lots of ATP via the electron transport chain in the mitochondria. Now, why do we make ATP? Why do we make ATP? We make ATP only so that we can break it down. only as a temporary storage so that we can later use it to do cellular work. And so this breaking down of ATP is going to be the opposite. It's going to be a hydrolysis reaction. We're going to have ATP. We're going to have to insert water here, cleave that third phosphate off, and we're going to end up on the right side of the equation with ADP, the existing phosphate no longer attached, and then we're going to release or liberate some energy. This is really the magic of breaking down ATP. The energy that we liberate or release, that is what we take to do cellular work. And when I say cellular work, I mean all of the jobs that our cells have to do. They have to transport things around. They have to make hormones, make proteins, make enzymes. They have to transcribe DNA, translate these proteins, right? They got a lot of cellular work, right? Machinery to operate, to make. They have to repair damaged DNA, they have to repair organelles like the mitochondria. And so there's a lot of things that have to happen on a cellular level that require this energy that is being stored in ATP and that can be liberated by breaking it down. Okay, so again, going back to that analogy of being that temporary backup charger, that we can now use it to charge our phone because it has the energy that we have captured there. And so the reaction that represents this process or represents glucose oxidation is written here on the screen, right? So we've got carbohydrates here plus oxygen, right? And this carbohydrate is in the form of glucose plus our oxygen. And on the other side of our equation, we're going to get carbon dioxide as a byproduct. We're going to get water again, because this is a hydrolysis reaction. And then we're going to get the energy that we have liberated. So the central reaction for breaking down glucose is written here. And the delta E, right, the energy change is about negative 686 kilocalories per mole, all right? So this is basically representing how we take food in. Let's say you had a piece of bread for breakfast this morning. You brought it into your GI tract. You broke it down into smaller pieces, which is going to be the digestion part. You absorbed it across your GI tract, which is the absorption part. And then you're going to take those macromolecules, which is now going to be glucose, which is the unit of that carbohydrate that you ate. And you're going to try to liberate the energy that's captured in that molecule of glucose. And how much energy are you going to liberate? This is that delta E. Okay. Now, if we were to, for fun's sake, think about this reaction in the opposite direction, can anyone think about what that would represent? Because we know that these reactions can occur bidirectionally, right? So if we had this reaction occurring in the opposite direction, can anyone think about that process and what that would be called? So if we take carbon dioxide, water, and energy, and we make glucose and oxygen, what is that process? Excellent, thank you. Yes, that is absolutely correct. So that would be photosynthesis. So photosynthesis is how plants can conduct this exact reaction in the opposite direction. They're going to take the sun's energy, which is thermal energy. They're going to take water and carbon dioxide, and they're going to give off glucose, which is what we can eat from plant sources, as well as oxygen, which is what plants release. And so notice how, in a very unique way, plants and humans, or most animal species for that matter, have this symbiotic relationship where we can give off what they need, which is carbon dioxide as a byproduct, water as a byproduct, and energy. And they can give off what we need, which is glucose, right? This is how we get our energy, and oxygen, right? Oxygen as our main source of cellular respiration. So this is really a great way to illustrate the symbiotic relationship between plants and animal species, and how this glucose oxidation can occur in the opposite direction as well. All right. And so that is a really important point to make here. Now, if we look at this illustration again, more of a graphical illustration. So we've got carbon or glucose here, oxygen. We've got glucose oxidation taking place to give us carbon dioxide and water. Now, the energy that we liberate is what we take and phosphorylate ADP in order to make ATP. Okay, and this is specifically going to be the oxidative phosphorylation, right? The phosphorylation that requires oxygen. That ATP is going to be made in this phase, but also temporarily stored. And then it can also be broken down on this phase, liberate that energy, and we can take that energy to do cellular work, right? Cellular movement, molecular synthesis, transport across the cell membrane, all of these roles within the cell that require the energy liberated from the ATP. What I want to emphasize here, though, is that this is not a perfectly efficient process, okay? This is not a perfectly efficient process. A lot of the energy that we liberate when we actually break down glucose, as well as when we break down ATP, is actually lost as heat. Okay, this is going to be lost as heat into the environment. And so this is a really important way that the body can generate heat, heat that is important for our thermal survival, but also heat that is just given off and lost into the environment. I'm sure if we were all sitting in a really small room, after a while, we would begin to notice that the temperature in that room would slowly begin to increase. And that is simply because of the thermogenic heat that is liberated from our body just by a nature of cellular respiration, of us breaking down glucose, making ATP, breaking down that ATP and generating the energy there. But not all of the energy that is captured and liberated is translated into cellular work. A lot of that energy, as we'll see, is going to be lost as heat. Okay, so here again is that equation. Here we've got the exact numbers as far as a single molecule of glucose. So for every single molecule of glucose, we're going to need 6 oxygens. And in doing so, we're going to generate 38 ADP. That's going to be phosphorylated by 38 phosphates. And on the other end of our equation, we're going to generate 6 carbon dioxides, 6 water, and then 38 ATP in total will be made. Now, this number can vary quite a bit. Really important to notice that depending on your body state, right? If you're elderly, this number can go down. You can be less efficient. If you're younger, a child, this number can go up and you can make much more ATP. If you're pregnant, if you're sick, if you're obese, there can be a lot of variation in this number. And so you'll see different numbers in different textbooks and different literature. And the range that you'll typically see is anywhere from 34 to 38 ATP, okay? 34 to 38 ATP, and that is, again, just based upon natural variation. Now, just to illustrate that this is not a perfectly efficient process, we're going to look at what would happen if we would take 38 ATP and try to work in the reverse, right? If we took 38 molecules of ATP, we would generate 38 moles by 7 kilocalories per mole, and that would give us 266 kilocalories of energy. So in theory, 38 ATP should give us about, or in our body will give us about 266 kilocalories per mole. So this is impractical, right? This is what happens in our body. Now in theory, 38 ATP should give us 686 kilocalories per mole, right? We looked at that initial reaction for glucose oxidation. And so in theoretical studies, this should give us 686 kilocalories per mole. But in our bodies, practically, we only get 266 kilocalories. So there's a really large discrepancy between how much energy is stored in 38 ATP and how much we actually liberate in our physiology. And the difference is about 420 kilocalories, right? Negative 420 kilocalories. So we say that to say that of the 686 kilocalories that we should ideally be liberating from 38 ATP, we only liberate 266. And so that means that glucose oxidation is only 38.8% efficient. Okay, and 61% of that energy is basically lost into the environment as heat, right, as our body heat. So that's a really interesting phenomenon to ponder, right? We ingest all of this food, we intake in all of this potential energy, and we only capture about 38% of that energy to do things within our cell. And the majority of that, more than half of that energy, is basically lost back into the environment.